to replace the motor neurons that have perished in the disease. There's significant developments being made in stem cell biology, but it has not become mainstream that one transplants a motor neuron and that it goes and makes the connection, the proper connection from the spinal cord to the muscle, and has the proper connections in the spinal cord to function like a brand new motor neuron. And I think there's still a lot of developments, a lot of work to do. There's certainly a lot of excitement in stem cells, and we share in that excitement of stem cells on the promise and the advances that are being made in producing motor neurons, on the potential to transplant them, and the potential to coax them to go out and make the proper connections. And certainly there's exciting work being done. Our feelings are that this is not at prime time, that motor neurons can be replaced functionally. That is, a new motor neuron is placed and it grows out to the muscle, makes the proper connections, and will start functioning.
Okay. Rosie McClay asks, well, I like a lot of the other questions being asked. I am very interested in the possible therapeutic benefits for older, larger children, whether they are type 1, type 2, or type 3. It seems to me when I heard him speak in June that he was having success in larger primates. It would be good to know the progress since then.
So thanks for your question, Rosie. Indeed, we have continued to make progress in those non-human primate studies. That is, we are confident that we are targeting motor neurons in older, or at least 3-year-old non-human primates. And we still have a bit more work to do, but our results have held up quite consistently. That if one delivers a marker gene through a marker gene that's inside of our virus, that this virus is getting to motor neurons, targeting those motor neurons, and those motor neurons are expressing the protein. So now we're in the stages of looking for that safety to move forward. I think we've addressed the questions of how old of a patient can one go to see or not see effects, and those questions really can be addressed by new animal models that are being developed, or potential new animal models that are being developed, and then human clinical trials.
Okay. Another question from Rosie is, will you partner with one of the SMA docs, like Dr. Sabota, to choose which children are enrolled in clinic and to establish the inclusion-exclusion criteria? Where will clinic take place? At multiple locations?
So we are quite connected, our basic research team as well as our clinical team, to a number of outstanding neurologists throughout the country and throughout the world. So we're certainly taking advice and guidance from the field on our developments. So yes, we certainly do have the interest for partnerships with a number of neurologists to move this study forward. Likely, the first clinical trial will actually happen at Nationwide Children's Hospital in Columbus, Ohio, and this will be led by my center director, Jerry Mendel, working together with John Kissel and many others to move this forward for the first phase 1 clinical trials. As far as inclusion-exclusion criteria, these are really things that go into the investigational new pre-IND package, and then ultimately into the investigational new drug application. And this is something where there will be certainly a lot of discussions amongst the neurologists, and certainly discussions with the Food and Drug Administration about what is acceptable, what is not acceptable for moving into human trials.
Okay. Jacqueline Tansey asks, does Dr. Kaspar think there is promise in gene therapy for all affected age groups?
So again, this brings up that all-important question of will this therapy work for my child? Will this therapy work a year from now for my child? The truthful answer to this is no one knows that answer. Again, what we do know is when we deliver the gene earlier, one has the greatest effects in the mouse model of the disease, the mouse model that normally survives 15 days. We're hoping it's important to help answer this question with newly models that are being developed, and then the human clinical trials will give us insight as we move forward to that, as far as moving forward into multiple patient populations of the disease.
Okay. Jacqueline asks another question. She says, I'm interested in knowing if Dr. Kaspar believes gene therapy will be a permanent solution, or if the affected individual would have to continue getting treatments for the rest of their lives.
So our gene delivery utilizing ano-associated virus is a one-time gene delivery. It's been tested in humans before, not for SMA to date, but in other diseases such as hemophilia. Muscle injections of the ano-associated virus has led to very long-term gene expression. So it appears that if the cells still exist and they've been delivered the gene, those cells continuously will produce the protein. So we believe it's a one-time gene delivery to deliver AAV vectors, to deliver our gene delivery for SMN. There's a number of other exciting developments in oligonucleotide therapies to increase the level of SMN. And those look like it would be more than one treatment. Now, there's a potential added safety to the oligonucleotides. That is, if there are safety considerations, one can stop giving the drug is cleared out, and one doesn't have to worry about anything. That's an advantage for the oligonucleotides in comparison to gene delivery. Once we put the gene delivery, it's there, and it's there for the rest of the lifetime or the rest of the lifetime of the cells that it targets. So there's balances, and there's good and bad for all of these therapies. One always has to weigh the opportunities, the risks, and the benefits in the design of any therapies. So our gene therapy, that's why we raised the bar quite high on the safety prior to moving to human clinical trials to truly demonstrate the safety of a gene therapy.
Okay, Jacqueline also asks, and I think many other parents you'll see will be asking the same question, how far off does Dr. Kaspar believe he is from human clinical trials? Who would be the first eligible individuals, type 1, type 2, or type 3?
So the timeline of a translational program, we've certainly made significant progress on the preclinical aspects. That is, we have repeated multiple times, as well as in outside groups, have shown that gene delivery of SMN in severe models of the disease have a significant extension of lifespan. The safety studies to get a handle on our safety prior to going into really expensive formal toxicology studies that are sanctioned by the Food and Drug Administration are getting off the ground, and we're starting to collect data to demonstrate, at least initially, that our gene delivery is safe and well tolerated. Once we have that meeting with the Food and Drug Administration, the timeline starts getting to be more solid. Right now, it's a bit hard to nail down an exact timeline, because we haven't had that meeting with the Food and Drug Administration where we go back and forth on discussions of what is it going to take to move into human clinical trials. Typically, a timeline, once you have that pre-IND meeting and one sets up a safety study, one can imagine 12 to 18 months. 12 is very quick, but it's between 12 to 24 months. One can think about moving forward, or certainly having a lot more data that one has confidence that the therapy is safe and can be moving forward to the human clinical trials. Now, the safety studies cost a lot of money, and then one has to be thinking about scaling up in a manner that the virus is produced for human clinical trials. That is good manufacturing practice production of the virus. Basically, this is what any drug is produced that provides additional safety, additional controls to ensure that we know that we're delivering exactly the gene and that it's past very stringent levels to be able to go into a patient. Those things cost money and they take time to develop. So that's why there's a timeline here of a kind of an open timeline, because this is really dictated based upon the safety that is produced in the formal toxicology and biodistribution studies. We don't want to move forward too quickly and run into problems in a patient. So if we can utilize those formal safety studies to dictate the timeline and work with the Food and Drug Administration, we move forward with what we believe is a safe and well-tolerated product that has the greatest potential to have a therapeutic effect in human trials.
And I think the same question is being repeated by Diane Catalano-Blair and Katie Pitson. So just on that, just to highlight on that, your theory is that since the beginning of your program, you have focused on rather robust safety studies. Is that correct?
That is correct. Once we found those the preclinical efficacy that we were seeing in the mouse studies, we've been working almost night and day to really advance this in a translational program to get a handle. Again, we raised the bar really high on the aspects that if our therapy, if our drug is going to cause a problem, let's figure out how to correct that problem on safety and toxicology, or stop the program and start doing other research. So we don't want to move forward with something that is going to be toxic or cannot enter into human clinical trials. Again, one never has any assurance that a drug is going to be 100% safe and well tolerated. In fact, no drug is 100% safe and well tolerated. This is an experimental drug. It is a gene therapy, and we need to have confidence, at least from our preclinical and our formal toxicology studies, that this is safe and we can move forward with it. If not, we need to go and start working on something else, go back to the drawing boards and either figure out the problem or start looking for something else.
So Katie Pitson asks, is your program still on track from previous conversations, and have there been any side effects yet?
So we believe our program is right on track. We're very I'm fortunate to have an excellent group working with me, working for me on the studies that we're performing to date. So we have generated a significant amount of data, a significant amount of confidence in our preclinical efficacy and our initial safety studies, and we're moving forward on the aspects of gearing up for the production of those safety studies and for the production of good manufacturing practice virus that would be moving forward towards the formal toxicology studies and eventually into the into human clinical trials if everything continues to move along quite well. So we're thinking into the future and doing things now that are along the tracks of moving towards phase 1 clinical trials. And then the second part of your question
was, have there been any side effects yet?
So in the mouse studies that we've performed looking, and we've looked quite hard for safety, any safety. Now, these are small studies, but we have found no problems with these mice. We've looked at all of their organs. We're sectioning those organs. To date, we haven't found any problems. Their serum chemistries have come back normal. Now, this is a mouse. We're getting ready to test it in non-human primates. So far, so good.
That's very encouraging. Where does the gene used for the replacement come from? Very good question.
So we have cloned the human SMN